Good afternoon, everyone, and thank you for joining us today in our expansion microscopy user group. Today we have Dr. Izzy Jayasinghe from UNSW, and she will be telling us a bit of what she's done in expansion. Also, reminder, I put in the chat that Google document, please put up your name so we can quantify like attendance and stuff. So I'll leave it up to you.
Thank you, Gabriela. I hope you can hear me okay. Brilliant. Thank you for the invitation to speak to the group, and it's nice to reconnect because this is a group that I used to be a part of back in the UK. And I'd like to start by acknowledging the traditional owners of the lands from which I connect, the Ventricular people, yeah, just grateful for the opportunity to be here and reconnect with the Australian colleagues after an absence of about 10 years. So the work that I'll present largely comes from the work done in my team by these three researchers, Dr. Tom Sheard, Taylor Shakespeare, and Rajpinder Sira. So just acknowledging them up front for all the amazing work that they have done over the past few years. Now, my group is, as with most sort of bioimaging groups, is interested in answering specific questions, research questions, and but just wanted to highlight upfront that we are in a number of collaborations, and it's been an amazing few years applying this technology to visualizing a whole host of different types of samples, and they range from size and scale from the macro to sort of nano scale. So these are some examples. This is a sea urchin larvae in the sort of more complex stage of development, and we often use expansion microscopy to sort of see the finer structures inside, and especially how they remodel in different environmental conditions. We use expansion microscopy also to look at sort of more traditional cell types. These are, on this occasion, the chromosomes and the proteomes that surround the chromosomes during cell division. We look at adherent cell types and sort of their internal compartments. Zebrafish, C. elegans, the sort of more macroscopic organisms, all the way now down to single proteins. And I will spend a fair bit of time talking about these proteins. These are ryanodine receptors, ion channels. And our PET system, I guess scientifically, what we're interested in muscle and striated muscle, cardiac muscle cells that are, that make up the myocardium and the skeletal muscle. And as far as cells go, these are larger types of cells, tens of microns in thickness, and an expansion microscopy has really played a part in making super resolution imaging of these really accessible. So I'll outline some of the specific questions that we asked with these technologies before going into some of the new method development work that we've done more recently. So in muscle, we're interested in calcium signaling, and you can kind of you can simplify these locations of calcium signaling to structures that look a bit like this. These are basically regions where the cell membrane, plasma membrane, comes in close contact with sort of termini of the ER. And one of the key proteins that we're interested in is this giant intracellular calcium channel called the ryanodine receptor. It's a large protein, about 2.2 megadaltons in size as a tetramer, and it's a bit square when you look at it from the top, about 27 nanometers on each side. And this is a calcium-sensitive calcium channel, which means that the way they cluster in the working cell determines a big part of how they work. And this is really the motivation for our imaging. And, understanding how they cluster in working cells and how the clusters change in diseases really is a big part of that work. Now, our early understanding came from early work done by biochemists who reconstituted these channels into lipid bilayers, and with EM characterized how they self-assembled into these clusters. But hopefully I will convince you with some of the images that I'll show that when we do in situ imaging, the organization is slightly different, and there's more complexity to that. So to give the sort of TLDR, the short version of the backstory to getting into expansion microscopy, over the past sort of decade and a half, I have been part of teams, some at various stages based in New Zealand and then the UK, incrementally improving the resolution at which we can visualize these. So, in the early 2000s, sort of diffraction limited imaging, showed this as puncta. When STORM and DNA paint came along, we could see sort of stepwise increments in the level of detail. And when DNA paint in its implementation allowed us to see what we sort of deduced as single ryanodine receptor channels labeled with antibodies. And that, showed a departure from that sort of crystalline lattice pattern that we had seen previously, in vitro. But there was a criticism that was always squared against us, and that was that whether we might be biased by our perspective. And the reason for that was that a lot of these clusters that I showed in the previous slide came from the sort of just beneath the cell surface, and they were often flat arrays of, or what we thought were flat arrays of clusters, and essentially STORM and DNA paint localization microscopy primarily is a 2D imaging method. You can do 3D implementations of it, but the question was whether we were able to capture the full 3D complexity of it. And this is very true when it comes to striated muscle, because if you look at ryanodine receptors shown in green here, this is a cross-section of a cardiac muscle cell. A lot of those clusters are actually in the middle of the cell. There's some that are at the edge, but the vast majority is in the middle. And they wrap around, these clusters are often wrapped around these tubes and membrane called T-tubules. So these are not flat arrays of channels. And the problem is, from an imaging point of view, if you were to image optically, let's say these are confocal and effective D-STORM point spread functions shown to scale against this sort of EM. If you blur that structure with this kind of blurring function, you're going to lose a lot of that curvature information, or this sort of three-dimensionality to that. And that was really the problem presented to us. So if there's anyone in the room who hasn't already been introduced to expansion microscopy, and I really doubt this because I was speaking to the expansion microscopy user group. I'm going to show you very quickly what it is. This is a method where you start with standard fluorescence labeling, of the targets that you might be interested in, and you and that first step, you basically do a functionalization, and then you polymerize an acrylamide gel both inside and outside of the sample. And then the penultimate step is that you perform a digestion or a denaturation that essentially unlocks the mechanical structures that hold that cell or sample together. And the final step is that you add water, which displaces sodium from the hydrogel and occupies the pores of the gel in a way that it expands. And it can expand, depending on the chemistry that you choose, by a factor of 1,000 in volume or more. And then you can put it on a conventional fluorescence microscope or some kind of super resolution microscope to really image that structure, now physically expanded. Here's a visual sort of illustration of what gel expansion is. Two gels that sort of started life in a tube like this, but obviously one has been expanded by hydrating, and this is the principle that we're following. Now, in this talk, I'm not going to talk too much about the chemistry, but this is just to show that these are two two other recipes that we use commonly. One allows a linear expansion factor of four times, and the other one allows a 10-fold expansion. The main difference is that here we're trying to polymerize a gel that is highly branched, but you can achieve higher levels of expansion by mixing and matching protocols. There are versions called iterative expansion and so on. So the idea is to try and distend and inflate that sample as much as you can. Now, when we started out these experiments, we tried both 4 times and extend expansion microscopy. And here's sort of the illustration of where this benchmarking went. We were interested in these banded structures called, in muscle, they're called Z lines, and they're often a lattice, this kind of 6-fold lattice that at each band that is occupied by a protein called alpha-actinin. And we can antibody label them, and we can image it with different modalities, and it's essentially a very nice intrinsic ruler. And with sort of conventional localization microscopy, you can see the improvement in the level of detail. And what we found was that with expansion microscopy, combined with Eriscan, we could start to match the level of detail that we could see with DNA paint, and particularly this sort of grooved pattern. And I'll show you later on some explanation about what this groove relates to. But it was nice to see that fine detail really emerging with a very different technique. And what was really exciting about expansion microscopy was that it's actually a true 3D super resolution method, because if you implement STORM either through high-low illumination or 3D localization, you're still going to get a fairly skewed or fairly elongated sort of localization function in the Z dimension, while in expansion microscopy you're essentially isotropically squishing the point spread function in plane and in axial dimensions. And in simulations, what we found was that this was a reason that we could see this level of detail that was previously limited to DNA paint that other techniques couldn't. And that was a crucial observation. Applying this to imaging ryanodine receptor, we saw this sort of, with DNA paint, near the cell surface, we saw this sort of braille-like pattern, single channel positions, and it was really nice to see that pattern being recreated with expansion microscopy being a different way of imaging. But unlike before, we could now go further in. So previously, imaging further into cells, DSTORM was sort of our benchmark. We could see those curved nanodomains, those curved clusters, and we could still see the individual protein localizations within it. And if you do 3D imaging as stacks, you can actually start to reconstruct different orientations, sort of these clusters sort of wrapping around tubes, and really visualize that sort of with surface rendering. What makes expansion microscopy, I think, exciting also is that it is a 3D imaging method. So what you can also do is that you can apply the same techniques to tissue section. So this is a cardiac muscle tissue section with a cell section in cross section, and so the red hot it shows sort of ryanodine receptors, and you can start to reconstruct how those clusters may be wrapping around those membrane tubes that we mentioned, and that was really satisfying to get to that sort of 3D kind of visualization with these methods. So as a sort of group that works in the sort of biomedical space, there's always a push towards applying this to answering some useful questions. And one of the questions that we were really interested in was, the mechanisms of right ventricular heart failure. It's the weakening of the right side of the heart. Often the wall of the heart gets thinner, and if you are unfortunate enough to live in a place like the UK, there's actually no real viable treatment. And we could simulate this disease in rats with an injection, and they go into right ventricular failure in sort of 3 weeks after an injection. And there's, one of the hallmarks is this sort of dysfunctional calcium signaling, and we wanted to look at what happens to these ryanodine receptors and the sort of domains that wrap around those transverse tubules, membrane tubules. And what was really exciting or interesting was that we could see, as with previous reports of other types of heart failure, that extra resolution that we could get in 3D could show us this sort of scattered approach, scattered organization of these channels compared to the kind of control. And this sort of helped to sort of describe the first sort of mechanism or morphological changes as sort of unraveling of this of these clusters, almost like a fraying of that structure. I'm going to move quickly now on to kind of methods, because I think this is what everyone's largely interested in. And there was a few lessons that we learned along the way, and few observations, few developments that came along that we kind of engaged with. When we first, sort of around the time that we published that work, one of the big developments that sort of blew everyone's mind was the idea that you can use expansion microscopy in combination with sort of generic nondescript sort of dyes to acquire images that show the full structure of the cell, that sort of match the not match, but approach the level of detail that you can see with sort of thin section EM. These techniques, have been described in sort of terms like pan-EXM and techniques like that. The idea was that you could apply these highly reactive fluorescent ester versions of fluorescent dyes, and they would go and react with any available amines, and essentially would give you a map of the density of proteins that is in that sample. So these two images are of HeLa cells, and the resolution is not quite as high as in the EM, but I guess you started to see some details that conventional fluorescence imaging wasn't available or able to show. And it leverages this idea that the concept of decrowding, which happens implicitly in the process of digesting a sample in the expansion microscopy protocol, the spaces that are made available and the processes that are kind of unraveled in the process that are amenable to being stained. And when we started to engage with this, what we observed was that depending on the dye that you're using, it would highlight different structures. So this is an example of nuclear sort of nucleolar structures being highlighted with specific dyes, and this one showed a very extensive ER network, and this one showed what we presumed were Golgi, and so it emerged this idea that maybe different dye species, could be used to give different weighting to cellular structures. And one of the real possibilities that emerged from this was the idea that maybe it could work as counterstains, and certainly other groups had shown that these NHS ester dyes can be used as a sort of a background canvas, if you like, to showing the context that of the antibody labeling that you can use. So this is an example of two antibodies being used, ATP5A1, which is a mitochondrial label, and GM130, I believe, stains the Golgi, used in conjunction with sort of an Alexa488 NHS ester. And I think this is still perhaps the most useful application of it, but we wanted to dig further, and we had a hypothesis, and it wasn't our hypothesis, it was something that was put forward by various groups, that maybe the differential weighting that you give to different compartments really emerges from the physical properties of the dye. And one of the physical properties that was described that way was the hydrophobicity, or water solubility, which could be loosely calculated, predicted based on the molecular formula of each dye molecule. And so we went on an investigation where we took 14 dye species, NHS ester and other types of ester species that you can buy off the shelf, that had varying hydrophobicity levels, and also spectral, emission or excitation spectra. And one of the broad observations that we made, and we struggled to kind of nail this down as a rule, but in human HeLa cells and cell lines like RPE1 and HEX cells, hydrophobic dyes tended to sort of label sort of the extremities of a cell and label the nuclear environment or the perinuclear environment less well, while the hydrophilic ones tended to give a very strong overall appearance and a very strong staining of the nucleus. Again, it was a broad trend that we struggled to sort of make a rule out of, because there were so many caveats. And one of the issues was it really depended on the stage of the protocol in which you apply the dye. So you can, if you're labeling with 2 NHS dyes, then it has another impact as well, that they can interact with each other. And here's an example where we compare, AZ488, which is a kind of a slightly evolved version of an Alexa 488, compared with 2 very similar dyes, but they're different partners. When you apply it simultaneously with AZ647, it gives you very strong sort of ER-like stain, but that ER-like staining is de-emphasized if you pair it with ATA647N applied at the same time. And for some other dyes, or some in other contexts, dyes applied in sequence can also impact it. So this is an example where it's paired with the same dye, but the sequence has been swapped, and you get different emphasis of the structures. So here the nuclei are very strongly stained, and here it's sort of de-emphasized. So the take home from this exercise was, yes, these 14 dyes are great and available, and you can strategically pick them to stay in different compartments, but you have to do a number of validations. You have to validate it against the structures that you want to emphasize. You need to validate it against the labeling patterns that you get when you combine dyes, and then against, also you need to standardize your labeling protocol. So, and what is the labeling sequence? At what point of the expansion pipeline do you apply the dye, for example? And this project, it actually was quite a stressful one, because it was very hard to nail down the rules, if you like. And that brings us nicely to the concept of validating expansion microscopy images, because this is something that has, as a technique, this has emerged in a very short period of time and been taken up by groups who were not previously interested in super resolution microscopy. So it's really exciting to see this unlocking super resolution imaging. When we started to do this, we were quite naive, but we had a system that was built into our samples that was a bit different. We were looking at muscle. So we had these intrinsic rulers in the form of sarcomeres, which are very stereotypic. There is a bit of variation, but one thing we could work out is that in a gel that has muscle cells oriented in different directions, you can ask, well, is the observed sarcomere spacing independent of the angle? If there was a dependence on the angle, you can say, well, there's a skew generally towards this direction. And so that was quite useful to work out that there's some level of isotropy and, spatially in the way it expanded. The other thing we could do was look at structures that were very precise and hard coded into the biological sample, and we looked at these double-banded fractalin structures and work out that spacing, and does that measurement of a measured expansion factor match the kind of expansion factor that we deduced from measuring the gel. And that was comparing those two measurements was actually quite useful for working out, okay, there's no kind of mismatch in the different scales. Another approach that we have taken is to simulate the structures that we're interested in and asking, can you, given the, let's say in the optimistic scenario, if this is the level of resolution that we can achieve, this is how much by which the point spread function is actually shrunk in the sample, can we simulate and observe the structures that we're seeing? So, these are those curved clusters of ryanodine receptor. You could then, simulate it and work out, in the best case scenario and the worst case scenario, what is going to be the level of detail that we can see to actually detect those individual puncta. And so these two simulations, these two columns refer to 10 times and 4 times expansion. And it showed that 10 times expansion, unsurprisingly, is better at it, and that it was good enough for a given structure that we understand from EM that it was a useful technique. For looking at those fractalin bands, what we worked out very quickly was that that double-banded structure came from a labeling gradient, because it's a 6-layer lattice. When you apply antibodies, they tend to label the outsides really strongly and then gets weaker as you go inside. And when we did that and we simulated, the way antibodies bind into these targets, and then you convolve it by the effective point spread function, you can actually recreate the experimental data, which is in the top row, through simulated data. And I think that's an approach that we have taken of simulating different scenarios and working out which one confirms the data the best. It's not very scientific in some ways, but it is the closest you can get, I find. Now, one of the very common questions that I get asked when I give these talks is, how do you know that your gel is expanding isotropically? And do you see distortions? And the answer, the first one is you don't know unless you could look for it. And the second one is there are distortions. So these are some of the obvious ones. So this is an image of a cardiac muscle cell. Again, this sort of sarcomeric pattern, stained with NHS esters. This is a very normal, typical pattern that you'd see of the staining of the myofilaments organized into these sarcomeres. This is actually a section from the same 3D image stack at a different level, particularly the bottom surface of that gel, where that is all mangled. And this is not a natural structure. And it, in fact, is absent in unexpanded samples. Some of the other types of distortions you see are fracturing or cracking. You get sort of these it's almost like an earthquake happened. And then also warping or shear, where things are kind of skewed in one direction. These are obvious ones that you can observe, but just because you cannot observe them doesn't mean that there aren't any other types of distortions. And one of the things that unlocked closer examination of this for us was sort of trying to adapt 3D printable structures to our experiments. So this work we published at the end of last year. This is a 3D printed microplate. It's got the same standard footprint of 96 square plate with square wells, and so each well will hold a sample, and that sort of goes some way towards higher throughput. Although I would not call expansion microscopy a high throughput technique at this stage. I know that there are some exceptional demonstrations. And in each well, what we place is a kind of a jigsaw puzzle piece that works like a spacer. So you assemble the spacers and you cast the gel in a square in the middle, the center of the well, and then you release that gel once it's polymerized, and then you run through the kind of digestion steps and then hydration. And if it's gone well, it'll fill the square well, and you can at that point put that whole plate on a microscope and do some programmed tracking type of imaging. And one of the things that this unlocked for us was to be able to track cells before and after expansion. So this is an example of a small green cell here, the corner. If you have used, say, a recipe that allows fourfold expansion, you can predict where it's going to end up once it's expanded. So you can actually calculate that and go back to that location and acquire, obviously, a pre-expansion image at the beginning, and then acquire images of the same cells post-expansion. And this type of pairing allows you to compare both the sort of improvement in the resolution in the level of detail side by side, which is not often the case with expansion microscopy experiments. But it also allows you to register the two images and ask, well, how well do these structures match? And there's where you start to observe slight shifts in smaller organelles. Like often what we find is sort of membrane-bound organelles like nuclei or like structures within the nucleus. ER, for example, are very prone to this. And from that kind of analysis, you can actually get quantitative as well. So this is one of the common standards in expansion microscopy to measure or plot what we call the root mean square of the error, essentially a measure of the displacement in those registered images as a function of the length scale that you're looking at. And it allows you to identify length scales where the distortions are the worst. It just helps you inform the length scales in which there are distortions, and maybe to inform you to either steer clear of it or repeat it if you're doing quantitative measurements. The other thing that it allows you to do is to look at angular nature of any distortion. So this is one where there's no particular preferred angle, but here's an example of a cell that had a very preferred direction of that root mean square error, and that would be a red flag in terms of it would point to kind of a directional distortion. Just in the final couple of minutes, I just want to show some examples of two-color imaging with this method. So you can do pre-expansion imaging and then go back and acquire multicolor images. Using that, I mean, that's obviously where the interesting biology happens, comparing different channels. But from a distortion mapping point of view, those two channels cannot offer independent maps of distortion vectors. And often, if there's a region where there's not a lot of information that can be filled in the other channel, that could inform you, that can help really consolidate those distortion maps. And I think that's the true value of it, of combining multiple channels for it. And of course that, this lends to kind of an overlay and appreciation of the level of improvement in the resolution enabled with it. So if I were to sort of summarize these as sort of the advice that we would give based on our experience in the most humble possible way, is that if you're picking up expansion microscopy for the first time, pick a recipe and a method that matches the type of sample that you're imaging and the kind of measurements that you're looking to make. I think increasingly there's an expectation that you would validate your data, and that's not just in the spatial errors, but it's also sort of the stains and how they interact with each other. Pre and post expansion imaging, I know is challenging, and we have many debates in my team about how practical it is to do this, and colleagues who just don't want to do this on every experiment. And I recognize that it's already a long experiment. So strategically working out how do you validate your experiment, and do you have to do that for every image, is a conversation that needs to happen on an ongoing basis. And I think when you're making measurements, consider the length scales or the errors that it imposes, and always approach it from a level of skepticism. If you see structures that appear to disappear once you expand, ask what is happening. And if you cannot really reproduce those morphologies with other types of imaging methods, then again, be careful in putting those forward. So that's where we're at. I guess it's an ongoing conversation because this is a very new technique. But hopefully, yeah, you'll be interested to learn how everyone else does it. And finally, to acknowledge everyone who has been a part of this, again, Tom, Taylor, and Raj, and then others in my team, and numerous collaborators who are actually now interested in working with us and applying this to their samples. And thank you for listening.
Okay, so that looks great. We'll open the floor for questions. If anyone has a question, they can type it in the chat. I think we already have one. So it's asking, what software did you use for the pre-post tracking? And if you can give a bit of detail about how.
So it's again, we haven't done a lot of work in automating this, but basically the approach is that when we acquire images, we tend to zero the stage to maybe the corner of that square well, and then we, when we acquire an image pre-expansion, we record the coordinates for that location from the stage readout. We actually put a very simple formula in our recent paper, if you want to go and look back at it. And it's a simple conversion, it's trigonometry conversion from basically triangulating from the location where you acquire the pre-expansion image to the post-expansion image. And then you can, once you have re-zeroed the stage to the corner of the well, once you have the post-expansion sample, it'll predict where you need to move the stage to find the same cell. That will get your sample to that ballpark. And the advice always is to try and acquire a bigger image than you need for your analysis or your publication. But it is still challenging. It is still challenging for someone who is starting to learn this. And we found this out, especially in the process of developing this work to publication. One of the things that we did was we repeated the imaging of the same cells at different time points of the expansion protocol. And so we were imaging these structures at least 3 times. And often, yeah, the 3 images are not complete. There will be an overlapping region. But that, I think the general principle of returning to the same location and imaging is a good thing if you can master it. Certainly a bit of skill involved.
I wonder also related to that, you need some particular things on the stage, like to be sort of calibrated in a way that you can rely on something.
That's right, yeah.
Another question, if there is different errors in the different methods, so like 4 times, 10 times, and it seems difficult to compare any size of readout from expansion methods because some of them are 4, 10.
Yeah, so that's a very good question. The work that I have shown, if I can go back to these, for example, this is this work predominantly focuses on 4 times expansion, just because we were trying to fit that into a micro well. As you can imagine, if you go from a small region, a 10 times expansion, that's quite a large well. So we have done a few experiments on 10 times expansion. The errors tend to be accentuated as you go to higher and higher expansion factors, which is probably not surprising. But it also comes down to the material that you're using. So in adherent cultured cells, the expansion, the errors or distortions are relatively minor compared to tissue. So in this paper, we actually show some tissue expansion experiments as well, where you can get this sort of, well, you can get extra dimensions of this. So you can get systematic errors that are kind of shifted in very specific regions. So shear distortions. You can get things like tilting and warping, and those are much more prominent in tissues. And that's just comparing different samples with one technique. We have found through our experience of imaging muscle tissue, if you want to go and image muscle tissue, which becomes relevant, especially if you want to study human hearts, 4 times expansion is about as far as we would go, because 10 times expansion just has involves too many distortions, and often you have to do an extensive readjustment of the recipe to make it work, especially when there's extracellular matrix involved. So I hope I have answered the question.
Could you imprint a pattern on the gel in the spacer?
Absolutely. This is a technique that's already kind of it's not on the spacer, but there's a technique that's been described called gel map, which kind of uses micro patterning that can be imparted onto the gel at the point of the gelation. So that's been done. We can also look at imparting that